Okay, guys, so this is starting Chapter 6, The Visual System. Um, I like to start with this image. It's from Wired Magazine in 2002, and it shows you kind of the goal of why is it as psychologists we try to understand perceptual systems, particularly the visual system, so well. And part of it is because we've realized that we are so reliant on vision as humans that people who lose their sense of vision really tend to suffer. So if we can recreate vision, that would do a lot of psychological good for our society. So this is showing you some technology from the early 2000s that was developed that actually made these glasses with a photocell in the lens that were able to be wired into the occipital lobes of patients who lost their vision due to some kind of illness or traumatic injury. And then we'll finish this chapter by looking at where we are today with this kind of science. But of course, in order to do something like this, we have to really understand the organization of the visual system in the brain. So as we talk about different sense systems and our perception of those senses, there's some kind of basic vocab that I need you to know. So if we're talking about sensation, that is just our simple awareness due to the stimulation of a sense organ. Right, so that's just some kind of receptor on your body responding to some kind of stimulus from the environment. When we talk about perception, that is the organization, identification, and interpretation of a sensation in order to form a mental representation. Or in other words, that is what our brain creates as our experience of that stimulus that was detected at some receptor in your body. Okay, so um, without perception, we don't have that conscious awareness of the stimulation. And perceptual experiences of the same stimulus are going to vary across people. Then, how we are able to get from sensation to perception is this process of transduction. So this is when sensors of the body convert physical signals into neural signals. Right, so this is the process through which all of our sense organs are able to turn light waves, sound waves, pressure on the skin, changes in temperatures, into a neural signal that the brain can interpret. So with vision, vision has really been a conundrum for scientists to study because the input that we get for vision is really goofy. Right, so if you think about what actually gets sent back into the brain, we have visual input coming in through the eye. It's immediately distorted, meaning that its size has to be shrunk to fit through our pupils. Right, so it doesn't matter if we're on a mountaintop and looking over hundreds of miles of space, or if we're sitting 2 feet in front of our laptop screen, whatever our visual input is, has to be shrunk down to fit into your pupil in order to get into your eye. So we get this tiny distortion of the visual world. As soon as it enters your eye, it goes through the lens, which flips that object upside down. So we get this tiny, distorted, upside down and mirrored image on the retina. And even though the retina is a little bit round, if we're looking at the representation of a single object on the retina, it's on a relatively flat space. So the retina is more 2D than 3D. Yet, we have this really rich, dynamic visual perception of the world. So how our brain is able to take that really crappy input and recreate our visual experience is really quite amazing. So that's what we're going to be going through and learning about in this chapter. Alright, so let's start with just this concept of light. So light is necessary for vision. There are no species that can see in the dark, meaning that they have vision when there's no light present. But there are certainly differences in our sensitivity to light. So if any of you have cats, you've probably noticed that they can skulk around your house at night relatively easily because they have a very low threshold for the amount of light they need to be able to see. But if it were truly pitch black, if there was no source of light in a space whatsoever, a cat wouldn't be able to see anything. You still need light in order to stimulate the sense of vision. Right? So, of course, we're not talking about animals that can navigate space without vision, right? So, echolocation through bats and that sort of thing is something else, right? That's not what we would call vision. All right, so when we talk about light, there's basically two different ways that you can conceptualize what light is. You can think of it as photons of energy, which are little particles, or you can think of it as waves of electromagnetic radiation. Right, so basically you can use light as a waveform, or you can look at it as particles of matter, these photons. So out of all of the different electromagnetic radiations that are out there, humans are able to see light that falls between 380 to 750 nanometers in frequency. Okay, so this next graph is going to make that a little bit more concrete. So on the top where you see it going from gamma rays to AC circuits, that's every type of electromagnetic radiation that exists in our environment. Right? If we were in class right now, there would be X-rays and radio waves and all sorts of stuff flying around the classroom that we cannot sense, right? Because we have no receptors that respond to that kind of stimulus. In between ultraviolet rays and infrared rays is this small band of electromagnetic radiation that we call the visible light spectrum. Right? So this little white bar is being blown up below into the visible light spectrum that we see. So you can see its range from about 380 to about 750 nanometers in wavelengths. That just means how fast the waveforms are going. And on the slower end, that's where we see our purples and blues, and on the faster ends is where we see more oranges and reds. So if we look at this visual light spectrum in the idea of it being a waveform, the different aspects of the waveform tell us different things about the light. So the length of the waveform, which is the distance between peaks, tells us what the color is, right, what the hue is. So if there's a big distance between these peaks, meaning that's a slow waveform, it'll look bluer. And if there's a short distance, meaning it's a fast waveform, it'll look redder. The amplitude, which is the distance between the peak and a valley, is interpreted as brightness. So higher amplitude waveforms are brighter colors, lower amplitude waveforms are lighter colors. And then the purity is how uniform the waveform is. Right? So if it is a perfect sine wave like this one is, then that would be something that we would perceive as a highly saturated or a really rich color. We almost never encounter pure colors in our natural environment. We usually have some kind of combinations of waveforms, so we don't get this uniformity. We have some peaks that are higher amplitude than others, we get shifts in frequencies, and that's where we get kind of the mottled colors, right? Think of looking at a tree trunk and the bark. Right? You're not getting like a pure tone of brown out of that. You're getting all these different combinations of waveforms that create kind of that texturized color of bark. All right. So once light enters the eye, as I mentioned, we have this lens immediately behind our pupil, and its job is to focus light onto the retina. So there are a bunch of muscles around this lens that can alter its shape so that whatever it is we're looking at stays in focus on the retina. And this ability of the lens to change shape is called accommodation. Right? So the lens constantly has to accommodate to keep things in focus on the retina. Right? So that's just the process of adjusting the lens to bring images into focus. So basically the lens is surrounded by these little muscles called ciliary muscles. And if you can imagine, if you had a zoom lens on a camera and you were looking at something really far away, you would press that zoom in button, which extends the lens so that it moves closer to the thing you're taking a picture of. So if you are gazing at something far, far away, the lens in your eye gets pulled tight so that it's not as concave and the back of it moves closer to the front of your eye, closer towards the pupil. And that allows you to keep things that are far away in focus on your retina. Versus if you shift the focus of your eyes to something that's close to your face, then those muscles push on the lens to make it more concave so that the back of it moves further away from the pupil to, again, keep it focused on the retina. Okay, so this image is just to show you that lens here. It looks just like a contact lens. So if something's far away, this back end needs to pull up closer to the retina, or I'm sorry, closer to the pupil. And if you're looking at something really close, then it gets pushed by these little ciliary muscles, and that moves the back end further into the eye to keep things on focus when they hit the retina here. Right? So it's just like adjusting a projector on a projector screen if you are moving the distance of the projector around. All right. We also have a bunch of binocular cues that allow us to determine how far away something is when we're looking at it. So binocular just means that these cues are based on the fact that we have two eyes rather than one. And so our eyes have to converge a bit when we are looking at objects that are close to us. Right? So if we have high convergence on our eyes, meaning you're basically going a little cross-eyed, they're turning in towards each other, which is what converging means. Then if you have high convergence, then that means that you're looking at an object that's really close to you, versus if you have low convergence, meaning both of your eyes are pointed straight forward, that means you're looking at something that's far away. Right, so our brain can actually monitor the angle of our eyes and use that to infer the depth of objects. Right, so part of how it can recreate this 3D world is just by monitoring how much we have to turn our eyes in to look at something versus not. All right, another binocular cue is called binocular disparity. And so this is the difference between the images on the two retinas. So our eyes are set a couple centimeters apart. So even if they're both looking straight ahead at the same thing, the image that is going to be projected onto the left retina is going to be slightly offset by a couple centimeters from the image that is projected onto the right retina. So again, the brain can compare the overlap between the two images on your retinas, and if there is a high amount of disparity, meaning that there's not much overlap on the retinas, that means that you're looking at something far away, versus if there is not very much disparity, meaning that the images on the retina are overlapping a lot, that means that you're looking at something close. Right? So 3 ways that the brain can track depth by monitoring how much the lens is flexed versus not, monitoring the convergence of eyes, and by comparing the disparity of the images on your two retinas. Right, so in this image, you can imagine if we were looking at the flowers towards the front of the image, if we were really standing in a field looking at this scene, our eyes would converge a little bit to look at that flower, and there would be a lot of overlap or low disparity on our retinas. If instead we shifted our gaze to the mountain peaks, our eyes would not converge as much, right? They would be more pointed straight forward, and the amount of disparity on the two retinas would increase, meaning that the amount of overlap between what they saw would get smaller and smaller. So these principles of depth perception that we get just from the mechanisms of our eyeballs has been manipulated to create these things called magic eye images. These were really hip and cool when I was a kid in like the early 90s. I don't know if you guys have ever done them. But basically, you can train your eyes to look at this image not as a 2D image, but as an image that has depth to it. And if you do that, you will see a 3D image pop out. All right, so this one is of a great white shark. I'm not sure if you guys will be able to get yourselves to see it. I worked really hard at this when I was a kid, so I can see them automatically now. But if you don't believe me, YouTube magic eye images, and you can see tutorials on how to learn to see these things. Okay, so a lot of stuff is happening just from like how we move our eyes and the mechanisms within the eye. But once that light gets back to the retina, focused by the lens, we also have a lot going on at the retina. All right, so the retina is an interesting little structure. It's built inside out, meaning that the light that hits the layers of it that are towards the front of the eye are not where the receptors for light are. Right, so the light has to pass through all these layers of cells before it actually gets to receptors that it can interact with. There are two pathways for communication on the retina. There's what we call the vertical pathway, which goes from these receptors to what are called bipolar cells to retinal ganglion cells. I'm about to get to a diagram, so it'll help with these. We also have lateral communication across the retina, and these use horizontal and amacrine cells to basically communicate to neighboring cells around them. Okay, so let's look at some images to help with this. Okay, so this is a magnification of this little sliver of retina. So the part that's towards the front of your eye is this top part here with the yellow cells, and the part that's at the back of your retina is this bottom part where you see this pigment layers. So light has to filter through all of these different cells before it gets to this pigment layer of the retina. And here's where thinking about light as photons is useful. So these pigments are able to absorb photons of light, right? And they absorb different photons. So some respond to red lights, some respond to green lights, and some respond to blue lights. So if they absorb photons of light, they will start to change their shape, and that will cause these photoreceptor cells to start to fire action potentials. So for these photoreceptor cells, they have these ion channels on these ends that can interact with these pigment layers, and then they can send action potentials on up to these other cells. You notice there's two types of photoreceptors. There's cones and rods. They're just named after their shapes. So cones have this little pointy end, and rods are more square at the bottom. And they can account for our two types of vision. So we'll talk about those in more detail in a minute. So the photoreceptors send signals up the lateral pathway to the bipolar neurons. Bipolar neurons are basically neurons that have two axons, so they can communicate in both directions. They can pass the signal up to these yellow cells, which are called ganglion cells, right, or retinal ganglion cells. And these are the neurons that form the big optic nerve that goes in through the back of your eye socket and all the way back to your occipital lobe. Okay, so that's a lateral pathway. It means it goes straight up in a lateral direction and directly into the brain. The horizontal pathways that allow the photoreceptor cells to communicate with each other and allow these retinal ganglion cells to communicate with each other is accomplished through these horizontal and amacrine cells. And you can see that they basically, rather than connecting up across these layers, they connect across the same layer. Right, so this horizontal cell is connecting all these different rods and cones together so they can communicate with each other, and these amacrine cells are connecting different retinal ganglion cells together so they can communicate with each other. So information can go straight up and into the brain, but it also can be passed across the retina. A couple interesting features of the retina is where that optic nerve leaves the eye socket, it's all white matter there, right? It's all axons, so there's no receptors there to respond to light. So this creates a blind spot in each eye where light can hit the retina and there's nothing there to respond to it. So we won't notice that that light is there. I do have a handout that hopefully I'll remember to bring to class next week so you guys can experience your blind spots. But what's interesting is we're never consciously aware of the fact that each of our eyes has a blind spot in it because the blind spots have binocular disparity, meaning that where the blind spot is on your left eye doesn't match where the blind spot is on your right eye. So your brain can do what's called completion, which is basically taking the information for the blind spot in the left eye from what's hitting the right eye and just filling it in, completing the missing gap. Okay, so basically our brain is making a best guess of what information should be at that blind spot based on what it's detecting on the other retina. Right? So that's kind of our first example of how when we really dig into perception, we see that as the brain recreates the data, recreates the information it's getting from the world, it makes a lot of guesses. So our perception of what the world looks like does not necessarily represent what is in existence in reality. All right, so the blind spot's an interesting part of the retina, but the most important in the retina is the fovea. The fovea is at the center of the retina, and it's where we are able to do what's called high acuity vision. Acuity just refers to detail, right? So this is where we can do really high definition processing of light. So if you are looking directly at something, you're looking at it so that the light that is being reflected from it bounces into your eye and lands on your fovea. And that's what allows you to see it crisply and clearly and in high detail. The fovea has less light distortion than other parts of the retina because the lateral pathway on the retina is kind of pushed to the side. So there's not as many cells that light has to filter through before it can stimulate the pigment cells in the retina. And so we have this phenomenon we call foveation, which is our natural focusing of what we're attending to on the fovea, which just means that if we're paying attention to something, we look directly at it to make sure that it hits the fovea of our retinas. Right? If we look at something kind of out of the corner of our eye without looking directly at it, it's blurry, it's not as clear as when we look directly at something. Right? So if something's important, we're going to foveate, which just means look directly at it. All right, I'm going to back up to the last diagram so I can point out where the fovea is for you guys. So this little divot right there, where you see that the retina is not as thick, that is the fovea. You can see it's directly behind the pupil. Right? So as the lens focuses lights, it's trying to focus it onto the fovea. So you see it's like a teeny tiny portion of the retina that does our most detailed visual processing. And as we move out towards the periphery of the retina, that's where we get that peripheral vision that is not as clear and not as high detail. Okay, so let's talk about why that is. So we have basically two types of vision. We have vision that we use when there isn't a lot of light present, right? So vision that is sensitive to, that is very sensitive to the presence of light. So in layman's term, we could call this night vision. And then we also have our acuity vision, which is our ability to see details, right? So our vision is constantly a combination of these two things. So the cone and rods of our retina contribute to these two types of vision. So cones do what's called phototopic vision. That is our high acuity vision in good lighting and our color vision. So phototopic basically refers to light vision or day vision. Um, so this in diurnal animals, which are animals that are up when the sun is up, we will see that there are more cones than rods present in the center parts of their retinas. Right? So if we look at the fovea of humans, we'll see that it's 100% cones. Right? There's no rods at that point in the retina. The rods basically do the opposite, so they do what's called scotopic vision. This is high sensitivity vision that allows for low acuity vision in dim light. Right, so this is like when you get up to go to the bathroom at night and you don't want to turn lights on to blind yourself, but you're able to still kind of see the outline of your bed frame and the outline of the door frame. So you can get around without banging your shins, but you're not really seeing color, you're not seeing a lot of detail. So that vision is coming from your rods, right? It doesn't need a lot of light to stimulate them, but they also don't give you a lot of detail once they are stimulated. So these are more common in nocturnal animals that need to be able to respond to very low levels of light. So what contributes to this is how they are wired on the retina, right? So that horizontal versus lateral communication on the retina is different between the cones and rods. So on the top here we have the wiring for cones. It's called one-to-one wiring or low convergence wiring, which basically means that each individual cone cell is synapsed with an individual bipolar cell, which synapses with an individual retinal ganglion cell. So that every single photon of light that a cone detects gets directly communicated into the brain. All right, so you can think of this like a high definition television where you have all these tiny little pixels giving you each little bit of information. Rods, on the other hand, have many-to-one wiring, or what's called high convergence wiring. Right, so we can see this whole group of rods synapses onto a smaller number of bipolar cells, and all those bipolar cells synapse onto the same retinal ganglion cell. So this is low definition vision because any signal across all of these rods gets blurred together into one signal going into the brain. Right, so that's why your peripheral vision and your night vision are fuzzy, right? Because they literally have fuzzy processing built into the wiring for that system. All right. Oh, sorry, I already jumped the gun on this question. So now that we know that about cones and rods, we know that if the fovea is where we do the most detailed processing in the retina, we know that we're only going to find cones at the fovea. And as we move out from the fovea to the periphery of the retina, the percentage of cones decreases and the percentage of rods increases. So the other thing that's different between cones and rods is their spectral sensitivity. So that is the relative brightness of lights of the same intensity at different wavelengths that they respond to. Right, so the different hues of light that they that they respond to. So let's look at a graph for this too. So our rods that are doing nighttime scotopic vision, you can see that they are maximally responsive to wavelengths of about 500 nanometers, which is, kind of this bluish-green color, right? So that's why at nighttime, things that are in that color range might appear the brightest to you because the cells that you're using for vision during that time respond best to that range of the spectrum. Now, our cones that we use for phototopic vision, you see that they're maximally responsive to kind of this lime green yellow color. Right, so during daytime, you might notice things like grass, leaves on trees, that they might appear brightest to you as you're looking around. And that's because the cells that you're using to process that information are maximally responsive to that wavelength of light. Right, so this again is spectral sensitivity, so what part of the electromagnetic spectrum they're the most sensitive to. But what's interesting is you could have the same amount of yellow light present in an environment and the same amount of red light, and you would perceive the yellow light as brighter just because your cones respond to it more than they respond to red light. Right, so another example of how our perception of the world doesn't represent reality. We can take photocells and measure the intensity of different lights and prove scientifically that they're equal, but people will still perceive and report perceiving differences in their intensities based on these spectral sensitivities. Okay. So once we have stimulation of the rods and cones and information going into the brain on that optic nerve through the blind spot, it goes through the brain on a route that we call the retina geniculate striate pathway. So retina obviously refers to the retina. Geniculate refers to the thalamus. So the thalamus is made up of a bunch of nuclei, which are just bundles of neurons that work together. And so the geniculate part of the thalamus is where visual information passes. And then it goes to the occipital lobe, which here is called the striate. That's because if you stain occipital lobe tissue, it looks like it's striped, and striate literally means striped. So retinal geniculate striate pathway really just means retina thalamus occipital lobe pathway. So 90% of axons of those retinal ganglion cells that make up the optic nerve go through the brain on this pathway. They do cross over, so information from the left visual field of each eye projects to the right lateral geniculate nucleus of the thalamus, and things from the right visual field project to the left lateral geniculate nucleus. Right, so just like we know that our right arm is moved by our left motor cortex, vision flips and is processed on the opposite side of the brain as well. Right, so stuff from the right side of space is processed by the left occipital lobe, and stuff from the left side of space is processed by the right occipital lobe. So again, that lateral geniculate nucleus is part of the thalamus. And then that thalamus, that part of the thalamus projects to the primary visual cortex. Right, so that often gets marked as V1, and as I said above, also gets called striate cortex. All right, so here is a picture of this pathway. Okay, so here we have stuff you're seeing in your visual field, and it's getting projected onto the retinas. Right, so in the left and the right eye, everything that's on the right side of space gets projected onto the left side of the retinas. Okay? From there, it goes down the optic nerve to this area called the optic chiasm. Now, for the left retina, the stuff that's on the left stays on the left, goes to the lateral geniculate nucleus of the thalamus, and then back to the left occipital lobe. For the right eye, the stuff that's going to the left side of the retina crosses over at the optic chiasm and also goes to the left geniculate nucleus of the thalamus and onto the left occipital lobe. Right, so everything from the left side of space ends up on the left occipital lobe. Everything from the right side of space ends up on the right occipital lobe. Okay? So you can see another feature of the visual system here, where the darkest shades of these colors is showing the part that's hitting the fovea, right? So this is the part that's hitting all those cones where we do that detailed processing. And you can see, compared to these other parts of space, it's very small. But if we look at the cortex, we'll see that a huge chunk of cortical tissue is dedicated to processing that small part of light that hits the fovea. And then as we go out to the more peripheral parts of the retina, we'll see that less and less cortex is dedicated to those larger parts of space. Okay, so that is a phenomenon called cortical magnification, that if we're processing something in a more complex or detailed way, we're going to need more neurons to process that information. So even though the fovea is a tiny bit of the retina, it gets a huge chunk of primary visual cortex dedicated to it so that you can maintain that high detailed visual processing. Okay. If we look at how this primary visual cortex is organized, we will see that it has a predictable pattern that we call retinotopic organization. So information received at adjacent portions of the retina remain adjacent on the occipital lobe. Okay, so retinotopic literally just means a map of the retina. Oh, I got ahead of myself again. More cortex is devoted to areas of high acuity, like the disproportionate representation of sensitive body parts in somatosensory cortex. Right, so that idea of cortical magnification. So about a quarter of our primary visual cortex is dedicated to input from the fovea, even though it's just a tiny fraction of our entire retina. All right, so let's look at an example of this retinotopic organization. So that what I'm going to show you is data from some cats research that was done, where cats were put in a head immobilizer, so their heads were held still, and they were shown this stimulus. This stimulus happens to stimulate the visual system really well, because the visual system is designed to detect contrast. So when we have all these black and white checks, they create a lot of contrast, and the brain will really respond to that. It just so happens that if you stain neural tissue while it is processing something, the active neurons will suck up that stain, and you can see which ones were active by which ones absorb the stain. You do have to sacrifice an animal to do this, so this is some of that unfortunate animal research where the cats are basically looking at this. They are killed, and at the moment of their death, a stain is put on their occipital lobe so we can see what neurons were responding to the last thing they were seeing. But when they do this, this is what we see. So this is a cat's occipital lobe. It's a lot pointier than ours is. But what you basically see is the pattern that they were looking at is perfectly mapped onto the occipital lobe. Right? So the stain was picked up by these neurons. So if you can imagine that target shape they were looking at, if we wrapped it onto the half of the occipital lobe that we can see here, that's exactly what it would look like. So that's what I mean by a retinotopic map. Where these different points of light are hitting the retina are mapped onto where they are processed on the occipital lobe. Okay. So as I mentioned, even though we get a ton of light incoming onto our occipital lobe all the time, it is designed to respond to contrast. So when we're interpreting visual information, we're basically trying to distinguish one stimulus from another, right? So if the information on the retina is 2D, how are we able to tell that if I'm looking at the class, that the desk is in front of you and not like some part of your stomach, and that you are behind the desk and a separate object from it, because it's all smushed together on my retina. All right, so this ability to respond to contrast allows us to detect the borders of separate objects and be able to identify separate visual stimuli from each other. All right, so the visual system is designed to enhance contrast that exists in our environment, and responds really well to change, or really when we say contrast, that just means change, like where there's a change in the type of light that we're perceiving. So I can demonstrate this contrast enhancement to you by showing you an illusion that are called mock bands. So mock bands are non-existent stripes that the visual system creates to enhance the contrast and make edges easier to see. Right, so it shows us an example of contrast enhancement. So if I show you these color bars, and if I asked you if they were shaded or solid bars of color, most people would say that they're shaded, and that they appear to go from light to dark as you move from left to right over a single band. Okay? But if you put your hands on your screen right now and cover up the sides of each bar and just focus on one, you'll see that it's a solid color all the way through, that there's no shading on any of these bars. So your perception of that shading, of it going from light to dark as you move across it, is an illusion. It's a perception that's created by your brain, because your brain is adding extra contrast to these bars that does not exist in reality. But by doing that, it is making the boundaries of these bars of color really, really easy to detect for you. And so you'll easily be able to tell different shapes and different stimuli from each other because your brain is going to magnify the differences between them. All right. This next bit is a little bit complex, so I think I'm going to stop here, and we will finish up this lecture next week with explaining what's happening neurally that creates these mock bands, that creates this contrast enhancement. All right, guys, I will see you in